Greetings everyone. So welcome to the first technical webinar on image analysis and DUS examination. And the program for today will have two parts. The first part with presentations, a second part with a session on questions and answers. For the first part on presentations, we'll have an introduction on UPOV guidance, which will be followed by the practical experience of UPOV members in China, Japan, Slovakia, France, United Kingdom. We will run the same webinar twice during the course of today. The next one within some 6 hours time from now. And to set the scene, a short introduction on UPOV guidance, in particular in document TGP-8, where we have a particular section devoted to examining characteristics using image analysis. The main benefit of UPOV is cooperation. And cooperation is enabled on the basis of a harmonized approach for conducting examination of distinctness, uniformity, and stability. This is what enables cooperation, and in particular the exchange of test reports. Also, that is linked to the strength of the UPOV system, where it generates variety descriptions at the end of the US examination that are internationally recognized. In this regard, document TGP-8, which is our document on trial design and techniques used in the examination of the US, which is also available on our website, you have there the web link, brings in section 11, guidance on examining characteristics using image analysis. The first criteria or principle set out in document TGP-8 is that image analysis is one of the possible methods for examining a characteristic. And just to recall, we're talking about characteristics that meet the basic criteria for their use in the US examination. Now, let's take a moment to look what are these basic criteria for characteristics to be used in the US examination. These criteria are established in the general introduction, document TG1, which is currently in its version 3. The characteristics are there described as the basis for DUS testing, and to bring characteristics to the US testing, they should be characteristics that result from the plant genotype, and that is excluding the environmental effects such as when influenced by temperature or light conditions. Therefore, standardizing these conditions may enable to assess the expression of the genotype. Also, the characteristics should be sufficiently consistent and repeatable in a particular environment. That is, that the characteristic should be able to be expressed if, for example, the testing is conducted in 2 growing trials or even in different seasons. In addition, characteristics for the US should express sufficient variation between varieties to enable establishing distinctness. We can easily imagine soybean flowers where you have white petals or purple petals. A DUS characteristic should be capable of being precisely defined and recognized. And this may be particularly relevant for image analysis, where you should be able to clearly explain what is the characteristic and define it as well. Every characteristic used for DUS examination should enable assessing uniformity. That is, the characteristic should be possibly observed in each plant within that variety, and also enable assessing stability, that is, over successive generations of cultivation, the expression of the characteristic could be assessed. Once again, document TG1 is available on the UPOV website under the TGP section or the guidance for DUS examination. Coming back to document TGP 8 in examining characteristics using image analysis, the second principle is that the characteristics to be assessed by image analysis should also be able to be examined by visual observation. That is, such as when a particular UPOV member does not possesses the machinery or does not want to assess the characteristic using an automated process such as image analysis, that still the characteristic could be observed visually or at least measured if the UPOV member wanted to. For harmonization purposes, whenever a characteristic is proposed for DUS examination, it should be clearly explained. And this is the case, for example, when the characteristic is included in the UPOV test guidelines, so that the explanations would enable to be clearly understood, that the characteristic is clearly understood by all DUS examiners. What you will hear over the course of this webinar is a number of criteria, and in particular, how different UPOV members control or address these criteria. For example, when recording images, there are calibration and standardization procedures that will be explained by each of the UPOV members in this webinar. For example, utilizing a same setup in terms of illumination, camera, the settings, the perspective of the images to be captured, and also the different calibration procedures to address, for example, the calibration of size, the illumination, and even to deal with light reflection, such as when you assess the color of a glossy or shiny fruit. And to the right-hand side of this image, you have the standards for calibration on color, for example, and also a tent that is used to provide indirect lighting. Finally, what you will hear over the next few presentations is linking to the practical experience of UPOV members in replacing what could be a manual measurement by a computer or an automated measurement. Please note during the presentations how many of these characteristics assessed using image analysis may require even more precise definition, so that you avoid introducing any biases. And also, these biases could be introduced by the genotypic expression of the characteristics, where certain varieties may express the characteristics slightly different than others, introducing biases that the computer may not be able to clarify itself, and thus requiring the expert advice. You will also see a diversity of software that is used for capturing and processing the images, as well as you will hear about the preparation of samples, how carefully prepared these samples are in order to be captured through image analysis, and also the different experiences in calibrating for assessment of sizes, colors, and the characteristics that will be here illustrated. All this guidance is available on the UPOV website under the section for DUS guidance. On this note, we would move on to the examples of image analysis in DUS examination provided by UPOV members. And I would like to invite our first speaker from China, Mr. Kun Yan. Please, Kun, you have the floor.
Yes, just now have some problem of the microphone. So thank you, Leontino. Dear participants, I'm very glad to introduce the experience of image analysis tools used in China. And I will introduce from three aspects. First, the history of development. Please, next slide. Next. This is the history of our development of image analysis. Actually, we start to use our own the first image analysis tools is DUSP. It's developed by a company, and actually we could push back to the history to 2002. At that time, we know this software, and during 2010 and 2016, we developed it as our want. And then we have the second one. It's almost in 2018. We buy another image analysis software. It's called SCG. It's also a commercial software. And since 2021, we developed our own image analysis software or algorithm, something like that. So next. And this is the interface and the components of the DUSP. It's including hardware and software. Next. Main features. First, it's sometimes it looks like a Photoshop, but have more measurement functions than Photoshop. So it could produce length, angle, area, diameter, number, and color. And we revised something for our own needs. We developed we put into all the shape and the color types in TGB 14 in this system, and a new title could be classified into one of them. And we also developed a new algorithm for similarity of two touches directly. But this software have a shortage. It needs a lot of hand operation, so it's low efficiency. And this shows the shape, how to do the shape. Only the whole shape need a hand choose, and all the other types could be analyzed automatically. Next. And this is the color analysis. It will accommodate 6 similar IHS codes by color distance. And we also have a good idea on analysis of color distribution, but still have something to improve. Next. Next. And this is show some other functions. Number analysis is a very basic function, and similarity analysis. This is what I mean. Put two live together, and we could get a similarity rates. And we also have an independent image database to manage all the data image data. Next. And SCG, this is a new software. Actually, it also include hardwares, and the advantage is one-click analysis. One click, you can get all the site data, including length, area, diameter, and the numbers. And it applies to maize, rice, wheat, barley, oilseed, vegetable, but mainly focus on the seeds. but it doesn't have many color analysis functions. Next. Here you can see it includes many hardwares, also include a scanner. Next. And this is an example for how to analyze a comb from the ear. And here it could produce the length and width, and also numbers of the corners, and also I don't know how to call that, the tip no sees area. They also could measure the length. Next. And this is another site to show how to measure the we call that back what we call it, how many lines of the corner, and also some width and length of the corner. Next. And this is our own algorithms related to the image analysis. We use some our old photos to we analyze some older photos using the image analysis algorithms. But at that time we found some problem. We don't have a good ruler to calibrate the size. So next. So later on we developed a ruler with 8 color circle. So by that we could easily to get the size and also the color calibration, and then we got the what we want. Next. We also have a very small software. We call it DUS. It's mainly used for data analysis, and inside we have a lot of functions to cite the photos to check, and then we use the photo to do some simple measurement. For example, we photo get the length from the ruler, then we can get any length of the patches, and also we could get the first click and second click two size data of color. Next. And with our new ruler, we under calibration algorithms, we could get the color also have the right color. So this right color could less than 20 RGB distance. Next. And this is some example. The upper, the left the left photos, one is too dark, another one is too light. And with our color calibration algorithm, we get the same color, we get all the color back. Next. Then now we are building our image analysis platform. It's completely with the data collect and analysis, and also get the final report. And in it, we use many ways to get the images or videos, and then after that some we could use it to check the result with the man, and also some photos we use to produce the data directly. So I think in future it could completely replace manual observation and measurement. And also it could enhance the precise of the result of DUS examination with the rich basic data of rich image data. Thank you. That's all of my presentation.
Thank you, Kun. We continue on to the next presentation this morning, and we would like to invite the colleagues from Japan. Please, Toshiya-san, you have the floor.
Yes. Can you hear my voice clearly?
Loud and clear.
Thank you. So, okay. Let me start my presentation at the morning session. So, good morning and afternoon and evening. I am Toshiya Kobayashi, a senior staff working for DUS Test Management Section of Center for Seeds and Seedlings, belonging to National Agriculture and Food Research Organization, NARO, of Japan. I would like to make a presentation about new techniques based on 3D image data for characteristic assessment as a UPOV webinar. Please, next. So, okay. First, let me explain the background and purpose of our attempt. The characteristic assessment conducted by NCSS is the basis of the plant variety registration system in Japan. And the result of these assessments are used to judge whether or not the candidate variety can be registered as a new variety. On the other hand, the characteristic assessment requires advanced observation and evaluation techniques. In addition, the current characteristic assessment involves time-consuming measurement of plant and photographing, as shown in these images. These measurement and photographing need to be concentrated in a defined short period of time, such as flowering season. Therefore, if plant images can be converted into 3D data and automatically analyzed, the advanced observation and assessment will be possible without depending on the experience of the staffs. And it is expected to shorten the time and reduce the labor required. We call this kind of 3D data as 3D model. Please, next. 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 So, as mentioned earlier, the goal is to conduct automatic analysis in the future. But before reaching that goal, we first worked on the survey in 2022 to verify whether it is possible to conduct a characteristic assessment by looking at the 3D model with the human eye. As a premise, a 3D model can be manipulated on a computer and viewed from various angles. Furthermore, it is characterized by a far greater amount of information than a flat photograph, as it reveals three-dimensional shapes such as unevenness. However, in general, it is said that 3D modeling, creating a 3D model of plants, is technically difficult. This is due to the plant's thin stem, thin leaves, and complicated structure. To solve this problem, the Research Center for Agriculture Robotics of the National Agriculture and Food Research Organization, NARO, has worked on developing a 3D modeling device specifically for plants. Thank you. Okay, next. This type of 3D modeling device was created in 2022 in cooperation with the Research Center for Agriculture Robotics in Japan. The plant was placed on center of revolving table and photographed with a camera from all 360-degree directions while being rotated little by little. In addition, the three-dimensional structure of the plant is reproduced on a computer by taking pictures from various angles with 4 cameras of different heights. once the setting has been made in advance, all that is required is to place the plant on the revolving table and press the start button to begin filming, which takes about 7 to 10 minutes. The computer is then activated for analysis, and the 3D model is completed in about 90 minutes. The entire process is completely automated. Okay, next. When photographing begins, the revolving table rotates and stops after every 5 degrees of movement. Then 4 cameras take images simultaneously. This is repeated 72 times for each sample, capturing images in all 360-degree directions. In the actual DUS test, 10 samples are assessed. But for this 3D modeling, one individual of each variety was used. Each variety takes about 7 to 10 minutes to take photos enough, and a total of 90 minutes to create a 3D model on a computer per variety. Okay, next. These are actual 3D model of chrysanthemum created in 2022. The reason why we are targeting the chrysanthemum is that there are the most applications for variety registration in Japan, and a lot of effort and time are required to assess the characteristic of the plant species. Thus, the measurements are displayed from the 3D model on the computer by manually specifying measurement areas such as diameter and height of the flower, height of the plant, length and width of the leaves, and the length of the petal. The 3D model on computer was also checked by a person, a human eye. We also checked the 3D model on the computer to see if they could evaluate the shape of the leaves and other characteristics as well as the real one. Okay. Here are 3D model created with a 3D modeling device in 2022. This is a flower of chrysanthemum. This is the 3D images. So it will rotate just for about 8 seconds. You can see that the 3D model reproduces the detailed shape and provides more information than the flat photographs. Okay, next. The result of our attempt in 2022 is below. First one is that we tested the prototype device to create 3D model in chrysanthemum. Second one is that we confirmed the size of flowers and leaves can be measured by 3D model we created. And third one is that we find out that we can get the equal result between 3D and physical assessment for 20 out of 52 characteristics in standard type of chrysanthemum of the DUS test. This means that 3D model can be utilized in process of assessment for certain characteristics. For example, petal attitude, flower head diameter, and so on. And 32 of the characteristics that could not be assessed in 2022 may be able to be assessed by creating a more detailed 3D model. We are conducting this attempt in 2023. These are our 2 challenges in 2023. The first one is to examine a close-up 3D modeling device to create 3D models that can distinguish even the small details of the plants. The second one is to expand the target plant species for 3D modeling to roses and dianthus. The result for 2023 are awaiting for publication still, and will be presented. So we'll make a presentation at the next opportunity. Okay, thank you for your attention. This is a photo for NCSS headquarters in Japan. So Japan is almost time for the cherry blossom to bloom. Okay, this is end of my presentation.
Thank you, Toshiya-san. We continue on to our following presentation by the experts from Slovakia. Please, Lumi, you have the floor.
Yes, thank you, Leontino. Dear colleagues, dear UPO members, observers, UPO staff, please let me introduce a brief presentation of the image analysis used in DUS examination in oilseed rapeseed in our examination office, Central Control and Testing Institute in Agriculture in Slovakia. We use image analysis to measure the characteristics of cotyledons and petals for oilseed rape. Based on the CPO technical protocol for oilseed rape, we have 4 characteristics that should be measured on cotyledons, namely their ratio length to width, saddle depth, ratio lobe separation to width, and ratio lobe separation to saddle depth. For each variety, please go back. Yes, thank you. For each variety, go back. Thank you. For each variety, the biggest cotyledon of 40 seedlings should be measured. As we have a huge volume of measurements for each year, the only possibility to get the measurements in time is to scan the cotyledons of each variety and to measure them by using the image analysis. Yes, of course, the cotyledons should be measured at the specific growth stage within a few days. It's very important. Next slide. The similar situation is with the characteristics for petals. As the flowering period of rapeseed is very short and is very often affected by weather conditions, we have to measure all 3 characteristics on each petal very quickly. Namely, we observe and measure length, width, and ratio of length to width. Next slide. For image analysis, we use the scanner with 18 megapixel resolution of images and 300 dpi. Here on this slide, you can see the process of the plant material placement as we use 24 petal leaves or cotyledons for each variety in 2 replications. They are scanned and saved as JPG image in the computer. When all varieties are scanned, images are processed by the ImageJ software. For that, we developed and optimized macros for measurements of all characteristics with cotyledons and petals. Please, next slide. During the years, we have collected some issues with the image processing. First of all, there was a problem with the size calibration of the images. Recalculation of the pixel size to millimeters, depending only on the 300 dpi resolution of the images. But we have observed that it's very insufficient, so we started to use the calibration disk. For us, it's a black circle on the blue background with the 3 holes, which means the disk diameter of 30 millimeters. With this way, we get more precise results in millimeters. Scanning the petals, there were problems with the positioning of the petals. They have very small leaves and they are very often moved from their positions. And of course, it's a plant material, so the petals are quickly very wilted and their edges are very often bended. To improve the quality of scanned images, we started to position the petals on the transparent film tape and then pass them on the paper. Please go back. This way we can prepare the sheets with petals at the flowering time and after scan them. Another issue is that many shades of yellow color of the petals, that the lightest colored petals had very low contrast to the white background paper. But the solution was quite simple. It should be used the complementary blue background paper. After that, we get the high contrasting scanned images of the petals. Please go back on the previous slide. Yes, the next slide, sorry. Yes, thank you. Here you can see examples of scanned images before and after image analysis processing. Results data are in Excel format and all measurements are in millimeters. We get one file for all images. Individual petals can be identified by variety name of the scanned image, replication, and by the unique number of that image. That we use for the next step, which is the checking of the ray data from the image analysis. Next slide. Our DUS experts, in cooperation with statisticians, check the images for outliers that shouldn't appear in the data for statistical analysis. We use for uniformity and distinctness evaluation of candidate varieties. As we can see on the slide, there are, for example, not correctly measured jaw distance on the cotyledon, as we can see on the image of cotyledon with number 314, or not correctly detected object outline, as it is on petal number 37, or cotyledon with number 312. Clean data are finally used for COI-U and COI-D analysis to evaluate candidate variety. After that, we used DUS software. Please, next slide. At the end, I would like to summarize some advantages of this image analysis for the DUS examination of oilseed rape. First, that we don't need to keep some exact positions for cotyledons and petals. For macro, we just need to keep enough space between objects and control only vertical middle line. It's for the reason that the cotyledons and petals should not be rotated. Positioning of the plant parts is less time consuming. In short time periods, we need to process samples for measurement. Putting the small petals on the transparent tape, they keep their shape and allows to process samples later by scanning. Not at the same time, but it's possible a day or two later. This reduces the time pressure on DUS experts in the busy period for collecting the plant samples. Biggest advantage is, of course, fast measurement and processing of all scanned images at one step. With that, the measures of petals and cotyledons petals. Next slide. Thank you. Our future challenge is to use image analysis also for measurement of rapeseed silks. Now we have these four characteristics as a length of peduncle, length between peduncle and beak, length of beak and width. We measure it manually by hands, but for the future, we have developed macro for image analysis. And for this season, we will start to test the measurements of these silks by the image analysis. And that's all from my side. Thank you very much for your time. And thank you for your attention.
Thank you, Lantino. Good afternoon, good morning, good evening, depending on where you are. My name is Clarisse Leclerc. I'm the head of the DUS department at CHEVES. GEVES is the French examination office in charge of DUS testing, also VCU testing of new varieties of different crops in the framework of PBR and also a national listing. So today I will present the experience of GEVES as regards image analysis in DUS examination. Thank you. So image analysis is used in routine for DUS testing in GEVES for more than 20 years now. So nothing new, I will present what is routinely used. It is used to replace visual observations or manual measurements on MS characteristics from the technical protocols or guidelines. So basically what is done as routine for DUS for many years is that we collect or harvest organs of different varieties, different plants in the field. It can be leaves, can be leaflets, can be petals, can be seeds harvested, can be pods, and so on. We have several examples, and I will display some examples afterwards. Once we collect, harvest the organs in the DUS trial, then we take back these organs in the lab or in the office. We scan or we take a picture in controlled conditions with definition of equipments and protocol defined. So it's very important to be in this controlled condition and to have defined a protocol as regards the equipment. And then we process the image analysis with a software that was developed internally. It's called AIM. It's a software that was developed together at GEVES with a statistician and IT experts. But it is based on a free software that some of you may know and may use, called ImageJ. So it's more like a software using this images free software. M is in the list of the software of UPOV document INF 16, as I put it below, since 2013. Yes. Next, thank you. So to give you some concrete examples how we use it in routine, I added a few slides about characteristics that are measured with image analysis at GEVES. So the first example deals with P. So we use image analysis to measure several characteristics at once on the stipule. So in the guideline, in the technical protocols, there are several characteristics to be measured on the stipule: the length, the width, the length from axil to tip, and the length of lobe below the axil. So to do that, as mentioned in the previous slide, we collect the stipule in the DUS trial. We take back the fresh stipule in a lab, and then we scan the stipule as in the picture on a scanner. It's always the same scanner with the same settings that we use for several years now. And then we process the image with our software. So we get an Excel file with the measurement, and we control the results of the scan with manual measurements of some stipule, but only a part, just to be sure that there is no problem with the automatic measurement. Next slide, please. Then there is another concrete example of what we do in routine. It's for flax we use the image analysis for the petal measurement. So in the guideline or in the technical protocol, for flax, we measure petal length and petal width. So to do that, we collect 20 fresh petals in the field. The petals are glued onto a self-adhesive paper. And then, once again, there is a scan that is performed, and the scanned image is analyzed by the software M, and we get this Excel file. On flax, another organ that we collect in the field is a seed. So in the protocol, we measure seed length, seed width, and the ratio. And it's quite a small organ, the seed of flax. So it's easier and more precise actually to do it with image analysis. So as for the petal, we process in the same way. We harvest the seeds in the field. We glue 20 seeds onto a self-adhesive paper, and then we scan the image, and we get the Excel file with the measurement from the software. Another example I wanted to share is the silic analysis for oilseed rape. It's more recent. It was developed only lately at GVS. It's quite also quite difficult to assess it manually, because we deal with quite small organs when we deal with the beak or the pedicel. Actually, in France, we only measure the ceiling length and the ceiling length of a beak, but there are several other characteristics on the cilic that can be measured according to the guideline. So for that, we also harvest in the DUS trial. It's 1 cilic on 30 plants per plot. We collect them in a bag, of course, properly labeled. That's, of course, a challenge. I didn't mention it before, but you have to properly label your samples when you take them back in the lab or in the office. Then the silics are placed on a blue paper. For silic, we use a blue, color blue paper. They are arranged vertically in 2 rows with the beak at the bottom. And then we take a picture in standard condition. Here it's not a scan, it's a picture. Also in standard condition, it means that we always use the same settings with the same camera. And then we process the image again with our M software, and we get an Excel file with our measurements. Something also really important is that we use a standard image to calculate the ratio between pixels and millimeters. So then we put in the software this ratio, pixels and millimeters, using the standard image. Usually we use a coin of €2. It's what we usually use in France. It's very important to calibrate the software. And so this was developed, as I said, quite recently. It's used in routine since 2021. It's the last organs we decided to tackle with image analysis instead of manual measurement. So that was some concrete examples, and here I wanted to put why, maybe some important thing, why we use image analysis instead of the manual measurement, and also important thing about the prerequisites, and also some limits. So, of course, you have to perform a good preparation of samples. We have existing procedures for each species, each organ we deal with careful calibration. Usually we use a coin of €2 and standardization when it's needed. So why do we use image analysis? We think it can help and provide more accurate measurements, a greater precision, especially for organs of small size, such as the petals or the seeds. And also, sometimes it helps when the characteristic is quite difficult to assess visually, for example, for ratio or for some parts of the stipule. It's not so easy to assess with the human eye. You can be distracted, you can say. And sometimes we think we have a greater precision with this analysis. And something also important is that we store images of plant material that we can use later for the description to illustrate the description file, but also we can process the image at a later stage also if we are in a peak time of observation and we have to do several things at the same time. We can store the image and process the image later. So now that we have developed this software, we are quite experienced for the routine, and we always try to extend to other organs or other species. It needs time to develop something and to check that we get the same results with manual measurements and with the image analysis. And so, well, this is currently developed also on some other species at GVS. And as for the limits, I wanted again to report something. So usually what we do is we collect the organ in the field, take back in the office or in a lab to take a picture or a scan. So it's not directly done in the field in routine, and sometimes taking back the organ, it's quite labor intensive. So that's one point. And if we do not see any advantage to do that, in addition, we still do the measurement manually in the field when it's possible. So far, this image analysis is routinely used at GVS for some characteristics like length, width, ratio, and shapes. But we have tried to do it for color assessment, but so far it's not used as routine, I have to say, I have to admit. But we are thinking about it, actually developing some R&D programs about color assessment using image analysis, but it's not in routine so far. And there is a last slide about this methodology, because we think there are also some opportunities of new vectors and new technologies. And here I put the example of some research we are doing internally at GVS about the color assessment of cabbage using a drone, a UAV flight drone, to help the examiners for outer leaf color, the intensity of color assessment. We also have tried it for the maize foliage intensity of green color, but so far it's only research. It's not in a routine, but it's interesting also to try to use these new vectors and also new technologies, maybe machine learning and so on. But it's for the future probably. And that was my final word for today. Thank you.
Thank you, Clarisse. I guess what you meant by future could be the TWM starting in a couple of weeks' time, but we will hold on to that. And meanwhile, I would like to invite the colleagues from Slovakia to introduce the next presentation on all-seed rape varieties.
Good afternoon, good morning, or good evening to everyone. My name is Veronika Bojdová, and I'm from the Central Control and Testing Institute in Agriculture in Slovakia. And let me introduce a brief presentation of the image analysis used in DUS examination of oil seed rapes in our institute. Next slide, please. We use image analysis to measure the characteristics of cotyledons and petal leaves of rape seeds. According to the CPVO test protocol and test guidelines for the rape seed, there are 4 characteristics that should be measured on cotyledons, namely the ratio of its length to its width, then the settle depth, the ratio of the lobe separation to its width, and the ratio of the lobe separation to the settle depth. For each variety, the biggest cotyledon of 40 seedlings should be measured. As we have a huge volume of measurements for each year, this year there are between 800 or 900 varieties. The only possibility is to get the measurements in time is to scan the cotyledons of each variety and then measure them by the image analysis. Because the cotyledons should be measured at a specific growth stage within a few days according to the weather conditions. Next slide, please. The similar situation is with the characteristics of petals. The flowering period of rapeseed is very short here in Slovakia, and it is often affected by weather conditions. We should measure these 3 characteristics on each petal, namely its length, its width, and the ratio of its length to its width. Next slide, please. For the image analysis, we use the scanner with 18 megapixels resolutions for images and 300 dpi resolution. Here you can see the process of the plant material placement. There are 24 petal leaves or cotyledons for each variety in 2 replications we need to measure, which are scanned and saved as a JPEG image. When all varieties are scanned, all images are then batch processed by the ImageJ software. We developed and optimized macros for the measurement of all these named characteristics of cotyledons and petals. Next slide, please. During the years, we met some issues with the image processing. First of all, there was a problem with the size calibration of the JPEG images. First, we used only a recalculation of the pixel size to millimeters based on the 300 dpi resolution of the images, but that was insufficient. So we started to use the calibration of the photographs. We use some coins first, but these years we use the calibration disc. There are some black circles on the blue background marked with holes, which mean the diameter of this calibration disc. You will see them on the next slide at the moment. This way we got the more precise results in millimeters. By scanning the petals, there were problems with the positioning of the petals. They are very small, and these leaves often moved from their positions. Petals were wilted quickly and their edges were bended. To improve the quality of scanned images, we started to position the petals on the transparent thin tape first, and then pass them on the sheet of paper. This way, we can prepare the sheets with petals at the flowering time and scan them afterwards in a few days. Another issue is with the many shades of yellow color of the petals. So the lightest colored petals had a very low contrast to the white background paper. The solution was quite simple. We should use some complementary color, that means the blue background paper sheet, to get the high contrasting scanned images of petals. Next slide, please. Here you can see examples of scanned images before and after the image analysis processing. The resulting data we get in Excel format, and all measurements are in millimeters units. We get one file for all images. Individual petals can be identified by the variety name of the scanned image and also by the unique number on that image. That is necessary for the next step, which is the checking or validating the raw data that we get from image analysis. On these images, you can also see the calibration discs. In the right upper corner of each image, there is a blue sheet of paper and calibration discs with 3 holes. So we used the 30 millimeter diameter calibration disc in these images. Next slide, please. Our DUS experts, in cooperation with statisticians, check the processed images for outliers, which should not appear in the data for statistical analysis used for uniformity and distinctness evaluation of candidate varieties. There can be, for example, as you can see on the images, not correctly measured the joule distance of the cotyledon. This is the image numbered 314. Or then there can be not correctly detected the object outline by the petal number 37, for example. The cleaned data are finally used for COU and COID procedures to evaluate the candidate varieties by the DUS software. Next slide, please. At the end, let me briefly summarize the advantages of the image analysis for the DUS examination of rapeseed. There is no need to keep some exact position scheme for the cotyledons or petals that we use for scanning. For the macro we use for image analysis, it is necessary only to keep enough space between objects, plant parts, and control the vertical middle line. That means that the cotyledons and petals should not be rotated before the scanning. So the positioning of the plant parts is less time consuming in the short time periods in which we need to process samples for measurement. Pasting the small petals on the transparent tape keeps their shape and allows us to process samples later by scanning, which reduces the time pressure of the DUS experts in the period of plant samples collection. And the biggest advantage is, of course, the fast measurement and processing of all scanned images at one step to get the measures of petals and cotyledons characteristics. Next slide, please. Our future challenge is to use image analysis also for the rapeseed calyx measurement. As we heard from colleague Clarice in a few moments before, they can measure the length of the beak and the length of the calyx, as I have mentioned her presentation. We have developed also a macro for ImageJ software and plan this year to test it if it is sufficient to measure all 4 characteristics of rapeseed calyx as they are proposed in the TGP and test protocol documents. Final slide, please. My time is out, so I would like to thank you for your attention.
Thank you so much, Veronica. We will continue now the webinar. The following presentation, we would like to invite colleagues from the United Kingdom. Please, Alex, you have the floor.
Sorry, I'm mute. Thank you, Leontino. Hi, my name's Alex Talabadine, and I work at NIAB as one of the technical managers for DUS testing. So yeah, I want to give a brief overview and some examples of how we use it in our testing. Next slide, please. So we have again also been using image analysis for a number of our tests for quite a number of years, and we've slowly developed it using it to take measurements for various species. But we find from our experiences that that it can be a really useful tool for reducing cost and time rather than you doing manual measurements. It aids consistency, as we've got can have multiple people preparing the samples, but then have a camera to carry out the single point of measurement. We find it's also very helpful for just reducing any risk of human error in manual inputting of data, and reduces the sort of dependency on expertise to carry out the measurements. By having the images, we also find that we are able then to trace back sample measurements if needed, and we have that as a sort of bank of data and to refer back to. Next slide, please. So just to give an example of what we use them for, winter oilseed rape, again, like the others, is where we use it the most. In any year, we collect over 2 million data points for 11 characteristics on the cotyledons, petals, and the silique measurements. We also use it within our field bean test and our test for sugar beet. Next slide, please. So again, very similar. Our setup is very studio image based. We use a sort of off-the-shelf DSLR camera setup with a stand and various lighting, using overhead LED lighting and underneath the backlit lighting. So depending on the plant parts that we're imaging, we'll have a different setup, and we have different calibrations and setups for each plant part. So in the examples below, we use a backlit system for getting the outline of the cotyledons. And again, like most others, we use a domestic coin as a standard reference object in this instance, but we also use a sort of black and white reference object for any images where we have a lighting overhead. We again have a similar setup using the blue card background for our petal measurements in oilseed rape, and we specifically use a matte coloured or matte sticky paper to reduce any glare or shininess that might affect the image. We also have a barcoding system that follows the sample from collection in the field to the image, and then that is used to then label the files, the images, and the data files that are then come out of the software that we use. And obviously, one of the advantages of doing it this way is you can obtain multiple measurements from multiple plants in a single image. The software we use also has a function to allow live object analysis feedback, so you can sort of draw on live outlines of the objects before you take an image and analyze it, in case there's any need to make any adjustments to the presentation. Okay, next slide, please. So I haven't covered what software we use, but we have a bespoke software that's been developed by a software developer, and it's based around using object libraries to build in the analysis, but it uses the concept of examining the pixels of an image and using the 2D geometry to determine measurements when there is a known reference object of a particular size. For the saliques in the example below, in oilseed rape, these again have their own specific preparation method where we use a sort of corrugated filter paper to ensure that the pods are aligned or should I say, that so that they lay flat. So when we're taking our width measurements, they're all in the correct orientation. And we use a calculation using radiuses along that midline to detect where the beak and the pedestal cutoff points are to automatically take those measurements. And yes, all the images are then, once collected, reviewed by the technical experts, and any measurements can then be manually adjusted if the calculation has not been successful. And at the same time, we will be scrutinizing the images for any obvious outliers or errors in the analysis. Next slide, please. So again, in this example, in field beans, we use the image analysis to take measurements from leaf samples, and in this example, it's not a characteristic that we use anymore, but we had used it to measure or how to say, determine the shape of the leaves, and we've also used in this example sort of algorithm calculations to reconstruct some of the leaf just to aid and use as a tool. So where there may have been significant pest damage, where we've got P&B and weevil maybe destroying a lot of the leaf, we built in a bit of a tool to try and reconstruct areas that were known to be damaged in case that was needed to take a true measurement of the length and width. But this is used specifically as a tool. We scrutinize this quite a bit if we are using that additional analysis. But it's just something that could be used. Next slide, please. So just looking forward to what other methods of image analysis we're developing. Again, trying to image in field has always been something that we've been looking at trying to do. Obviously, you lose maybe the standardized setup and controlled lighting conditions that you can have in a studio image. But we have been most recently trying to develop our software for a mobile application. And it's kind of at the minute there for us to try and play with. We're not using it for anything, but it has its own challenges where you're not using maybe a camera stand. So built-in analysis to try and correct images where you may be taking photos from an oblique angle, but we're aware that technology is moving on. Cameras and phones and the processing power that they hold could be quite useful in obtaining images in the field. The other advantage is you can have multiple users taking images with sort of minimal resources. And again, with how we work nowadays, using devices to capture data in the field, you've got the option to store data in the cloud and have things backed up. So there's the sort of peace of mind with collecting data nowadays. But that's all in development for a possibility of use. Next slide, please. So maybe a bit more advanced and somewhere where we're making a bit more progress is with using UAVs and drones to capture sort of full trial images. We've seen quite good correlation recently using drone images to sort of recreate 3D images of the trials and measure crop heights. And we've been investigating a lot into how those measurements correlate with our manual measurements, and for some species it's quite useful, and the data we're getting out of it is interesting. And again, like most people, we're very interested in trying to develop our assessment of color. We've tried many attempts in the past, but we're looking at using the RGB cameras on UAVs along with the multispectral cameras, as they're becoming a bit more affordable and easier to get hold of and to use in maybe assessing some of the vegetative indices and using it for green color assessments on a wider scale. Some of the benefits, obviously, of using these is you can capture data from a whole trial within a very small sort of window time frame. So you're capturing everything at the same time, which is particularly important maybe if you're assessing color in the field. Getting the lighting conditions consistent can be something that needs to be thought about. But obviously, it can be quite an efficient way of capturing a large amount of data, but there are limitations, and it's something, again, we were just exploring and seeing how it will fit into a routine DUS test. Next slide. I think that's me. Yeah. So thank you for listening. Thanks for having me.